How do you break down glucose? So one of the things, when I get to metabolic pathways, that students are always disappointed is that I never say, draw glycolysis. I'm not going to ask you to draw glycolysis because, first of all, I didn't ask you to memorize the structures and so forth, okay? But I will test your knowledge of glycolysis and pathways by questions that I ask. So you will need to know what I've told you about glycolysis at the level I've given it to you, okay? But that's the most common comment I get is, you didn't ask us to redo glycolysis. I'm not going to. Not going to happen, okay? But you will have questions that will be relevant to that you'll be asked on the exam. Okay? The exam format will be like the first two that you've seen. No big surprises there. Hopefully, I never want to have surprises on exams, but no big surprises there. This exam is getting into metabolism, and from the rest of the way out, we'll be doing pretty much metabolism the rest of the term, which only has a couple of weeks left. Good news there. And that's about all I have to say. So, questions for me? Yes?
Can you explain how in histidine tagging you can choose a protein to tag?
Sure. Yeah, good question. So her question is, can I explain how histidine tagging works so that you can tag a protein that you want to purify? Is that a reasonable way that you asked your question? Okay, well, let's go back and look at histidine tagging. Histidine tagging was probably right down under genetic. There it is, right there. Okay. Well, this shows how it works, but it doesn't get at the question that you asked, which is, how do I pick that protein? How do I get that protein that way? Okay. So how do I decide I'm going to do histidine tagging, and then how do I set this whole thing up? That's really her question. Okay. Let's imagine, if you will, that I have an interest in making a ton of human growth hormone. So the protein that I want to purify is human growth hormone. And let's imagine that I want a bacterium to make that protein for me. Okay? What would I need to do to make this happen? Well, what I would do would be, first of all, I would isolate the gene, the coding for the protein that I want. So I would go to the human genome. Oh, I hope that's not somebody waiting to get in here. Hopefully not. Okay. If they've got a class in here, then we'll have to move. I didn't even think about that. We'll see. All right. First of all, I have to have the gene, right? So I go to the human genome, I pull out the gene, I've got the coding for the protein, right? And I take that gene and I put it into an expression vector, right? So an expression vector has a promoter, it's a plasmid, right? It's got a promoter so that it's going to transcribe that gene. Everybody with me? But I don't just put that gene in there. I actually put a little thing at the very front of it that instead of starting with the gene, it starts the coding for histidine, 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 histidine. Okay? So when the bacterium transcribes that messenger RNA, it's going to have the gene, and it's going to have, as in the very front of that, coding for several histidines. Does that make sense? So the protein that's going to be made is going to be histidine, 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 and then the rest of the protein. Okay? So I have decided that this is the protein I want to get. So now my protein that I'm interested in purifying from this bacterium has histidines on it, and no other protein in the cell will have that, because the bacterial proteins won't have that histidine, or those histidines. I bust open the cells, and now I just pour that mixture onto the top. And so now I go through the rest of what I've talked about here. Before the cell replicated, that's correct. Right. And so at this point, then, the histidine tag proteins are going to stick to the nickel column, which is what's happening here. All the other proteins will come through, and then I just wash this with the equivalent of the nickel stuff. There's another trick that I use, but to get the proteins off. Okay? And I'm done. Josh, could you ask them if they're waiting on this classroom? If they are, we're going to have to move. Does that make sense? Other questions on that? They've got a review session. Damn. Okay. I'll tell you what. Let's follow me back to ALS, and I've got a classroom there we can grab. Okay. Sorry about that. I got to shut this down. Okay, so far so good. Sorry. Okay, you guys can get all your hard questions together while I log in here. That'll take a minute to come up. Sorry about that. I should have checked that the room was available. I didn't even think about it. So last two times, the last time I got away with it, I guess. So it doesn't happen all the time. All right. How you guys feeling about the exam? It's always a lot, yeah. Is it more than last time or not?
It's more like cycles and like things that it's not like specific different little things, but like one thing that with one part out you're screwed.
Okay. One of the good things, is like it says in the song, is that on the final exam you get a note card that you can bring that you fill out with everything that you want. So you've got that to look forward to, but you don't have that for this exam. So you get to plan it all in up here.
How long is your final exam?
It'll be, it's a good question. I usually don't make it twice as long. I usually make it about 1.5 times as long, but it'll be worth 200 points. Yeah. Actually, I shouldn't say that. I usually make it worth about 150 points. Then I scale it because in the calculation it's 200. I'm sorry? Same format, yep. Or I could make it a worse one if you'd like. No. What would be a worse format?
All just long answer questions.
All long answer? Oh.
I think your multiple choice.
You think so? Okay. Write a song. That's right. Okay.
Or you're asking us to get a canon.
Ooh. You really do have an evil mind there. Okay. Ah. Okay. All right. Now I think we're set. All right. Hit me. I should never say that in front of students, I suppose. Yes, ma'am.
Can you go over the different ribosomes, such as like S50, and then S50 or 16S, 50S?
Oh, okay. Sure. Sure. Yeah, okay. Good question. So her question has to do with the different nomenclature I've used to describe ribosomes and ribosomal RNAs. Okay? And what their function is. Okay, good. So let's go back to protein synthesis and talk a little bit about the ribosome itself. So first place, okay. This was a figure I showed you that showed you the sort of assembly of the complex that makes proteins. So the complex that makes proteins, I will remind you, consists of a ribosome, messenger RNA, and tRNAs. Okay? That's the complex that makes proteins. Ribosome, messenger RNAs, and tRNAs. The ribosome contains proteins, and it contains ribosomal RNAs. That's the rRNAs. So that means that in this complex, you've got all three forms of RNA: tRNA, messenger RNA, and ribosomal RNA. Everybody with me? Okay. Now, your first question was the different, what she called different ribosomes, the different components of the ribosome. All right? So there are two main components to the ribosome. They're called the large subunit and the small subunit. The large subunit has a name. It's called the 50S. 50S, like the letter S. And the small component has a smaller structure, and we call it the 30S. Okay? Both the 50S and the 30S contain ribosomal RNAs. Okay? And here's where they're found and what they are. The large subunit contains the 23S rRNA. That's the ribosomal RNA, the 23S. The 23S is the ribozyme. That's the thing that catalyzes the formation of the peptide bond. Okay. The 30S, the small subunit, contains the 16S ribosomal RNA. The 16S contains the Shine-Dalgarno sequence. And that's the sequence that forms the base pair with the messenger RNA. Now there's also another smaller ribosomal RNA I haven't talked about and I won't talk about here. And eukaryotes have a second small one, and we're not going to talk about it here. Okay? What does the Shine-Dalgarno sequence do? Okay, so good question. What does the Shine-Dalgarno sequence do? If we look, actually I'll show you here. If we look at this structure right here, we see here's the 30S. This is the small subunit we're looking at. And you can see that in this process of setting up for translation, the 30S has bound to the messenger RNA. How did it bind to the messenger RNA? It bound to it by virtue of the fact that the Shine-Dalgarno sequence in the 16S ribosomal RNA is forming base pairs with a part of the messenger RNA. That's what the Shine-Dalgarno sequence does. So the Shine-Dalgarno sequence forms a base pair region with the messenger RNA. And the purpose of that is to position the AUG in the right place. This positions the AUG exactly where it's supposed to be. Yes, ma'am?
So is there a name for the sequence or region in the messenger RNA that is binding to the Shine-Dalgarno? I thought
They're both called Shine-Dalgarnos.
Okay.
So you get, one's a complement of the other, but if you refer to either one of them as Shine-Dalgarno, I'll be happy. Okay? Does that help? Okay. I'm glad you asked that question because I probably wasn't as clear about that in class, and I talked around it, but I didn't give you explicit descriptions of what were there, what was there. So one of the sort of guidelines I would give you about things here is I think students sometimes don't focus as much on what I say as they should. And so I will always ask you questions about what I say in class. And that's why the highlights are helpful in terms of understanding what's there. And if you start with those as a good starting point for your studying, I think that's the best approach that you can have for doing things, because the highlights are what I remembered of what I said in class. And the highlights is what I use to make the exams. Okay? So focus on that. Focus on the things that I say. And yes, if you need additional things to understand what I say, then I encourage you to seek those out as well. But I'm not going to ask you things at any deeper level than what I say in class. Yes?
When you say expression vector, are you specifically referring to a plasmid with antibiotic
Yep.
resistance that's used for biotech? That's not really like a thing that we would just think of as like systematically.
Yes. So I'm going to repeat her question here, which is, when I use the term expression vector, basically what am I talking about? And is it specifically something like this, or is it something that occurs naturally? So an expression vector is a man-made plasmid that takes pieces of things that already exist in nature. So for example, you might say, oh my God, we've got antibiotic resistance. We're going to be having a problem. Well, the antibiotic resistant gene that we're taking is one that's already evolved in nature. We're not creating something new. We've taken that gene out of another plasmid that is found in bacteria. The replication origin, which is the second thing that's there, is obviously also present in nature, in plasmids and so forth. The third thing that I said that was found in expression vectors is a promoter. And again, we've stolen a promoter from a gene to make that happen. So everything that's in that expression vector was originally in nature. We just put them together into one package so that we can use those. Yes?
So would the expression vector be kind of like a, so to say, like Frankenstein plasmid then?
A Frankenstein plasmid? No, I wouldn't call it a Frankenstein plasmid. They're really not significantly different from plasmids that would exist in nature. With Frankenstein, I think of creating something that is totally different, that is bringing something to life that wasn't already alive. And I would say that's probably not an accurate way of saying that, yeah. Another thing I should point out, and this is something I usually say in class, and I didn't say it, but I'll say it here just again so people don't get concerned. We talk about antibiotic resistance, and we worried about penicillin not becoming effective and drugs not becoming effective. The bacteria that we use these with that have these things are all regulated and destroyed. They don't make it out into the environment. They don't make it out and spread antibiotic resistance. And moreover, the reason that we've taken these pieces from existing things is that the pieces that we've taken leave behind the parts that allow them to be transmitted. The reason antibiotic resistance moves throughout the bacterial world is because these genes that they have evolved, have evolved with transmission signals that allow them to move from one bacterium to another. We have taken those transmission signals off so that all that is there is the gene, and it can't go to another bacterium. That is, when this bacterium replicates, it can. But I mean, it can't say this bacterium bump into another bacterium in the environment and transmit its DNA. It can't do that. Okay? So that's a very important distinction in biotechnology that a lot of people don't really understand. It's important to understand that. And no, I'm not going to test you on that because I didn't talk about that in class, but that's just so you know. Okay? Yes?
How do viruses recognize their target cells?
Oh, good question. How do viruses recognize their target cells? Generally, they're recognizing a protein, but they could be recognizing a carbohydrate as well. And so what a virus will have is it will have a virus is has a coat of protein that's surrounding a nucleic acid. So we think of that coat of protein as protecting the nucleic acid, but that coat of protein also has a binding site for something specific for a cell. So it might be in the case of, I didn't mention it in class, but I'll mention it here. If we look at HIV, okay, HIV recognizes a protein that's on the surface of our immune cells called 4. I want to say HD4, but CD4. It recognizes CD4 that's on the surface of our immune cells. And by that protein being on our immune cells, it grabs a hold of that and now has entry to get into the cell. Okay? A skin cell doesn't have CD4 on it. So it's not going to grab that, and it's not going to get into a skin cell. Make sense? But they'll always have something specific for the cell that they will typically infect. And that's why, for example, we don't see viruses having usually a very wide range. Some viruses have a wider range. Flu virus has a fairly wide range. But it's also binding a more common structure on different cells so that it has a wider range because of that. Most viruses are fairly specific. So, for example, bacteriophages, which are viruses that infect bacteria, can't infect your cell. In fact, if they were to infect your cell, if you were to inject one into your cell, it couldn't do anything because it wouldn't have the right promoters and the right ways to make it stuff that it would do. So one of the things that is increasingly happening is one of the ways that the meat industry and other industries are getting rid of bacteria within the products that they're selling is to infect them with bacteriophage. Because it's not going to harm you, but it's certainly going to be nasty to the bacteria that are there. So that's just, yeah. Yes?
Can you go over elongation factor G and elongation factor T?
Yeah, EFG and EFTU. So we're talking about the process of elongation in translation. And just briefly go through those for you. So if I go to right here, and I go to the process of elongation, this actually figures in my book, so I've got to work. I can't complain about it. I used to complain about the books, various books figures, but this comes out of my book, so I've got to work on this figure. This is a bad figure, okay? It is. It's too small. All right. So what's happening? Let's first of all think about what's happening during elongation. During elongation, the polypeptide chain is growing because we're adding amino acids. And in order to add the right amino acids, the ribosome has to slide down the messenger RNA 3 bases at a time, 1 codon at a time. That's the 2 things that have to happen. Okay? So the first part of that process is getting the new amino acids into the protein. That requires transfer RNAs carrying the amino acids. And you remember I said that the bond between the amino acid and the transfer RNA is very unstable. And that means it has to be covered up by a protein. The protein that covers it up is called EFTU. EFTU covers up that bond and keeps it from getting exposed to water. So this EFTU is carrying or is binding to this transfer RNA amino acid complex. It lands in the ribosome, and it lands in the A site. And the anticodon of the transfer RNA tries to pair with the codon of the messenger RNA. Right? So you can imagine these trying to pair. If they are complementary, they're going to pair. Right? And if that happens, EFTU says, everything is fine, my work is done, and EFTU goes away. And if those two don't pair properly, EFTU recognizes that and says, we're out of here, and it grabs everything and floats away. So the function of EFTU is to protect the bond between the transfer RNA and the amino acid, and second, to see if that base pairing is occurring properly between the codon and the anticodon. Now, EFG's role is much simpler. EFG uses the energy of GTP to slide the ribosome down 3 bases. Moves it 1 codon away. That moving the ribosome down 3 bases is called translocation. And that's what the function of EFG is. Okay? Yes? Blue-white screening. Sure, sure. Since I said it backwards in class, yes. All right, let's get blue-white screening down for everybody. All right? Blue-white screening. And right here. Okay. First of all, when we think about introducing DNA into cells and DNA into plasmids, both of those processes are relatively inefficient. So the things that I've described to you, like antibiotic resistance and blue-white screening, are ways to ensure that we're getting the thing that we want efficiently. Okay? Now, I'll start with antibiotic resistance, and then I'm going to talk about blue-white screening. Antibiotic resistance is an important screening tool. Because if I've got a plasmid that I want to put into a cell, and I work to put it into a cell, the process is very simple, but the efficiency is very low. I might get 1 plasmid in 10,000 cells. And all I want are the cells that have the plasmid. I don't want the cells that don't have the plasmid because they're just going to make a mess for me. Okay? Well, I make the cells tell me which ones get the plasmid by selecting for antibiotic resistance contained on a gene on that plasmid, so that only cells that get that plasmid will be resistant to the antibiotic I put in the mixture. So if I put ampicillin in there, only cells that have an ampicillin resistance gene will grow. Everybody else will die. So the first part of it, which is getting the plasmid or getting the DNA into the cell, is very easy to take care of. Well, now I've got to step back a step and say, well, how did I get the DNA into the plasmid in the first place? I might want to put it into an expression vector. Maybe this thing I'm putting in is an expression vector that's got a gene, but how did I get the gene into the expression vector? Are you with me? So I'm going to draw on the board here a plasmid that I've cut. I'm just going to show 1 strand, but assume it's 2 strands there. All right? A plasmid, actually, no, I won't. I'll put 2 strands there. Okay? A plasmid that I've cut with a restriction enzyme. Okay? This is what it would look like. It'd be linear, it'd be whatever, but I draw it so it looks like it's a circle. And I want to put in a gene into here. Well, it might seem very simple. I just put DNA ligase with this mixture of these 2, and then this guy now combines with this guy, and I'm set. And that will happen to a limited frequency. The vast majority of ligations that will occur will not occur between 2 molecules, but it'll occur within 1 molecule. The most common thing that will happen is this guy will ligate this end to this end, and we won't get anything in. So I want to be able to tell which plasmids have an insert. Which one's got this? Most of them won't get this. Everybody with me so far? Okay. Now, how do I ensure that? Well, I use a different kind of selection, or a different kind of marker. We call them markers. The way that this is set up is so that this guy right here has a promoter for a gene, and it has a coding for the LAC-Z gene. So you see I've cut right in the middle of the LAC-Z gene. All right? If I ligate this back together, what's going to happen? I restore the LAC-Z gene to being completely functional. And if I put this in the middle of it, I interrupt the LAC-Z gene, and it's no longer functional. Right? So if I have a way of telling which cells have a functional LAC-Z gene and which cells don't have a functional LAC-Z gene, I know which ones have no insert and which ones have an insert. The ones that have an insert will be those that don't have a functional LAC-Z gene. Yes? Because the LAC-Z gene allows me to select for color. I'm getting to that. That was the part that wasn't covered previously clearly. Okay? Everybody understand? If it's got no functional LAC-Z gene, all right, it is going to have an insert. If it has a functional LAC-Z gene, it's not something I want because it just came back together. I took the piece apart, I put the pieces back together. Well, the LAC-Z gene is used to answer your question because the LAC-Z gene will cut a chemical that I can give to cells called X-gal, X-GAL. When LAC-Z is functional, it will cut X-gal and produce a blue color. So cells that grow that have a blue color will not be ones that I want. Okay? Cells that don't have a functional LAC-Z gene will not have that blue color. In fact, they will be white. So cells that are white have no functional LAC-Z gene and therefore have an insert. Does that make sense? Does that help? And I apologize for getting the blue and white backwards last time. That was embarrassing. You know, one of the things you discover as a professor is that your brain will often go in one direction, your mouth will go in another direction. And after I said it, I was like, that doesn't make sense. And then I realized, of course, it didn't make sense because I had the cleaving to make the white instead of the blue, and that was my mistake, so I apologize. Yeah, yes, question, yeah. Gibbs free energy, okay. So Gibbs free energy is the energy available to a cell to do useful things. Okay? It's the energy available to a cell to do useful things, right? So the Gibbs free energy equation is shown there on the screen. The delta G is equal to the delta G0 prime plus RT times the natural log of the concentration of products over reactants. What does that mean? Okay. Well, first of all, the Gibbs free energy, the change in the Gibbs free energy for a reaction tells us the direction of a reaction. It tells us the direction of a reaction. That is the change in the Gibbs free energy for a reaction. This part on the left, the delta G. If the delta G for a reaction is negative, the reaction goes forward as written. If the delta G for a reaction is positive, it goes backwards as written. And if the delta G for a reaction is 0, the reaction is at equilibrium, meaning it's not changing. Okay? No net change in forwards or backwards. Essentially, it means it's going forwards at the same rate it's going backwards. All right. Well, notice that the delta G is equal to the delta G0 prime, which is a different form of delta G, plus RT times the natural log of products over reactants. What in the heck is the delta G0 prime? Delta G0 prime is the standard Gibbs free energy. And it's the change in Gibbs free energy for a reaction under standard conditions. That's all it is. It's the change in the Gibbs free energy for a reaction under standard conditions. Everything at 1 molar. Okay. Starting with equal concentrations of everything. That's what that is. Well, reactions don't occur under standard conditions in our cells, right? So we want to know under the conditions in our cells. Well, that's why the delta G is useful, because it tells us under whatever conditions we happen to have. It tells us, okay, that as this ratio changes, delta G will change. Okay? Should get a pointer. See a pointer in here? I guess not. Okay? As the products and reactants change, the delta G will change. The delta G0 prime for a reaction is a constant. It doesn't change for that reaction because it's under standard conditions. Standard conditions are always the same. Okay? Now, the natural log term varies just like the log term varied. If the ratio is greater than 1, the log is positive. If the ratio is less than 1, the log is negative. If the ratio is equal to 1, the log is 0. Well, now this makes sense, because if you look at this equation, standard conditions means equal amounts of products and reactants. What happens to the log term? It's 0, right? And the delta G0 prime at that point equals the delta G, which is what I said. The delta G0 prime is the Gibbs free energy change under standard conditions. Okay? Don't confuse delta G0 prime with delta G. That's the most common mistake students make, is confusing those two terms. Okay? Let's say that I told you that I had a reaction that had twice as much products as reactants, and it had a positive delta G0 prime. Which direction is the reaction going to go? I hear backwards. How do you decide it's backwards?
The natural log of a number greater than 1 added to the delta G0 prime, then it's bigger than the delta G.
Okay, so if products and reactants is 2 to 1, that means this term is positive, right? And a positive plus a positive is what? It's got to be a positive. So the reaction's got to go backwards, right? Make sense? Let's say I've got a reaction that's at equilibrium, and there's 5 times as much reactants as products. Is the delta G0 prime positive or negative? That's correct. At equilibrium, when there's 5 times as much reactants as products, is the delta G0 prime positive or negative? 1 positive? Negative? 1 negative? Should we vote? How many positive? How many negative? Sorry, it's positive. Okay? Because if I have 5 times as much reactants as products, what's the value of the log term? Negative. If I'm at equilibrium, what's the value of delta G? So how do I get 0 if this is negative? The delta G0 prime has got to be positive. Right? Right? Everybody's frowning at me. Nobody's throwing darts. At least that's good news. That make sense? Now everybody's saying, yeah, okay. All right, good. Other questions? You're getting your head on this, right? The Vulcan mind meld. What's that? Not make that into one of the long answers? Maybe you think 40 points on that one or so? No partial credit? There's 1 vote for this. You two can duke it out. So, other questions? Yes, question in the back. Microarray. Okay. Microarray is a way of analyzing all of the messenger RNAs of a cell at the same time. That's what a microRNA is useful for. A microRNA is useful for measuring all of the RNAs of a cell simultaneously. Question? Yes, microarray. What did I say? Micro. Oh. Sorry. Thank you. Microarray. MicroRNA is something completely different. So it sounds alike, huh? I can't blame you if I can't say them right if you don't get them right, right? What's that?
What is it useful for? Measuring microarray.
Microarray. It's useful for measuring the levels of all the RNAs of a cell. A microarray. Okay? Now I've said it right. All right. What is a microarray? Okay, let's sit down. A microarray is something that we make in the laboratory. And we make it in an array. So everybody knows what an array is, right? An array is a sort of a set of distinct places, like a checkerboard is an array. If we took that checkerboard and we made the squares microscopic, we could put onto something that's about this big, we could put hundreds of thousands of little squares. Right? Little squares. Each little square is going to have one distinct thing on it in hundreds of copies. Hundreds of identical copies. What's that one thing? Well, I know from the human genome, or any genome, I know the sequence of all of the messenger RNAs. I already know them. Okay? So I can go to a machine. This sounds kind of magical, but it's true. I can go to a machine, and I can punch the sequence of a given messenger RNA into that machine, and that machine will use chemistry to synthesize the DNA that corresponds to that. Okay? So let's say I've got a gene for human growth hormone is my favorite, human growth hormone. I go to my machine, I punch in the DNA sequence of human growth hormone, and it chemically synthesizes one of those strands. The strand that's complementary to the messenger RNA for human growth hormone. All right? So I get hundreds of copies of that same sequence, and I chemically bond it to that first square on the microarray. Okay? And then I do that for every single gene in the human genome. 30,000 individual spots, each of which has hundreds of copies of each one. Right? So the first spot corresponds to human growth hormone. The second spot corresponds to insulin. The third spot corresponds to hemoglobin. Whatever way I want to organize them, I can organize them. Okay? So I've got an array. And I know where each of those spots is. So I could say hemoglobin in my computer program would take a microscope and it would go and it would look right at that individual spot where the hemoglobin DNAs are stuck. All right. So I've got my array. I then go to my cells. And I say, okay, I'm interested in examining liver cells. And I've got a liver cell that's got a tumor. And I've got a liver cell that's normal. I'm going to bust open the liver cell that's got the tumor. I'm going to isolate all of the messenger RNAs that are in there. And I'm going to attach a red dye to them. And I'm going to go over here to the normal liver cell. I'm going to bust it open. And I'm going to get all the messenger RNAs out of it. And I'm going to put a dye on them that's green. So all the messenger RNAs from the tumor cell are red. All the messenger RNAs from the normal cell are green. And I mix them. Okay? I've got Christmas, red and green, right? I mix them, and then I pour them onto my array. Swirl them around, and let them find their complementary base pair sequence. So they're only going to form base pairs with their complement that's on that array. I wash off the stuff that isn't stuck, and I take it to my microscope, and I start looking for colors. This is what I would see. Some of the places on the array will have no color. meaning that that gene was not made in either cell, either the normal liver cell or the cancerous liver cell. Some of those spots are going to be green, and those are going to correspond to the normal liver cell. The intensity of the green will tell me how much of that messenger RNA was being made. Intense green meaning a lot of things, a lot of green there, a lot of messenger RNA. A light green meaning very little. Some of the spots are going to be red. Those are going to correspond to the cancer messenger RNAs. And if I see green, it means it's being made only in the normal cell, not in the cancer cell. If I see red, it means it's being made only in the cancer cell, not in the normal cell. And if I see equal amounts being made in the two cells, I will see yellow, because red plus green in this array gives yellow. And there's a yellow. Well, I'm going to be very interested in what genes are being made in one but not the other, because it tells me this is a difference of a cancer cell. Some genes will be on in cancer cells and not in normal cells. Some genes will be off in cancer cells but not in normal cells. And some will be made in equal amounts. Some won't be made at all. But now I could look at this array and I could say, oh, okay, here are the genes, X, Y, Z. I could tell which ones are being made in the cancer cell but not the other cell, and vice versa. Make sense? People smiling with that one. Okay, good. It's a cool technique. It's a really cool technique. Yes?
Could we go over about 5 enzymes in glycolysis? Just briefly.
What the 5 enzymes are in glycolysis that I asked you to know? Okay.
Yeah.
Well, they're in the video.
I heard something about aldolase
Yes.
last Friday, but I didn't really hear about it today.
Well, I didn't give you the 5 enzymes today, right?
Okay.
So that you did hear about aldolase. Yes, that's correct. But I would recommend you go back and watch that video. That's why I make videos for you, right? Yes? Aldolase. A-L-D-O-L-A-S-E. So you should be able to draw glucose, fructose, ribose. You should also be able to draw sucrose. Did you find the 5 enzymes? Okay, so I'll repeat them if you'd like. So the 5 enzymes of glycolysis: hexokinase, PFK, aldolase, G3-PDH, and pyruvate kinase. The reason you didn't hear about all 5 of those today was because I talked about the 3 that are different in gluconeogenesis, replaced by 4 in gluconeogenesis compared to the 3 of glycolysis. Yes?
Does F26BP correspond with glycolysis only, or is also correspond with both glycolysis and gluconeogenesis?
Okay, so F26BP doesn't correspond to either pathway. It regulates enzymes in both pathways. When I say correspond to, it doesn't appear in either pathway. It's not made in glycolysis or in gluconeogenesis. But it regulates the pathways by binding to PFK and turning it on, or FBPase and turning it off. It's what we call a reciprocal regulator. Okay, guys, I think you're about out of questions, and my tummy is grumbling, so we don't want to think about that. So, okay. Good luck. I will be in a meeting in the morning from about 8:30 until about 9:30, but I think other than that, I should be around. So if you have questions, come see me.